This is a production of Cornell University.
Thank you for being here, and I'll get started. So, how many of you have heard of this proverb, we are what we eat, or you are what you eat? A lot of you, right? Okay. How many of you have heard of this one? Yeah, no one. Well, it doesn't exist. I actually made it up. But hopefully by the end of this talk, I'll convince you that it's not actually far-fetched. So, in a striking majority of the developing world, food systems, including those set out by the Green Revolution, have failed to provide the necessary forms and quantity of essential nutrients needed for optimal human health. So the Green Revolution was really successful in addressing the problems of food insecurity or hunger, but sort of overlooked the sort of, it's as important problem of nutrition security or malnutrition. So scientists have primarily focused on less sustainable methods and interventions such as fortification and supplementation, whereas intervention surrounding the soil, which forms the foundation of life, has been disproportionately lacking. So soil serves as the primary source of minerals to humans. It is both an indirect source via the food chain, and it's also a direct source through soil ingestion, inhalation, or dermal absorption. But the latter happens in such minute quantities, and it's also, for the case of soil ingestion, is socially awkward. Water is also another source of minerals, but it generally is low. Its supply is around less than 5% of the required total daily intake, with the exception of calcium and magnesium, which can provide up to 20%. Actually. Okay. So, as such, humans and livestock that monotonously consume locally derived diets subsisting on cereals grown on nutrient-depleted soils will often exhibit the same or a related nutrient deficiency, and this has been well established. For example, India produces enough food to feed its entire population, yet it has the highest prevalence of malnutrition in the world. And this can be attributed to two reasons, and it's partly attributed to the narrow focus on calorie-dense crops that the Green Revolution really championed in lieu of the nutritionally invigorating ones, and also because of the major depletion of the soil's nutrient reservoirs as a result of the continuous cultivation of the same technology. But we must not forget that a healthy life is not possible without healthy soils, as was promoted in the International Year of Soils. So what is soil health? Soil health is a concept. It's that looks at soil holistically, drawing away from the more traditional outlook on soils that are purely chemically driven, and emphasizes the less appreciated biological and physical attributes of a soil. In other words, it's the intersection of the soil's physical, biological, and chemical states that are anthropogenically influenced. So what we want to know is whether we can relate soil health to human health. In other words, are soil health constraints reflected on human nutritional status? So not much work has been done in this discipline, especially in recent years, and almost never on a household level. And it's partly because of the complex relationships that exist between soil and human health, which are difficult to extricate, and also because of the presence of many confounding factors. Some of the research that has been done includes one that was conducted in the Kishan disease belt of China. Kishan disease is a selenium deficiency disorder, and this study basically looked at the selenium distribution of in the soil, the grain, and the humans, and they found that it significantly decreased, the concentration significantly decreased with increasing incidence of Kishan disease. Another study conducted in two high prevalence of esophageal cancers in South Africa and Iran. And they found that the low levels of molybdenum and zinc in the soils were significantly associated with the prevalence of this cancer. A third one, also conducted in 2 agricultural regions of China known for their centenarian and nonagenarian residents, and it found a connection between the soils, the water, and the staples of those regions with this human health attribute. And the last example is a study conducted in tribal districts of Madhya Pradesh, India, and they found a strong interdependence of zinc content among soil, grain, and serum blood samples in this region. And this is well established, as you can see in this. So in this map, the areas with high zinc deficiency are co-located with the areas with high human zinc deficiency. So all these studies are, although extremely informative, their shortfalls lie in the fact that they only studied one mineral nutrient at a time. And they've been studied in isolation. The other shortfall is that they only focus on the soil chemical properties. And so didn't really look at the biological and the physical states. So that's why I decided to study the link between soil health as a whole and rice and human health. And I did that in Jharkhand, India. Jharkhand is a state located in the northeast part of India. Its population is 30% of its population is tribal, against the national level of 8%. They are resource poor, infrastructurally isolated subsistence rice farmers. 80% of the cropland is covered in intensively monocropped rain-fed rice. In India, rice contributes to approximately half the daily caloric intake, and it's presumed to be higher in Jharkhand. It also has a devastating health crisis. And it's considered one of the highest health burden states of India. It has the highest prevalence of underweight children, 45% child mortality due to severe malnutrition, gender-based food consumption disparity, where women often receive less nutrients, both in quantity and quality, because they're often eating the leftovers of their husbands and their children. 35% of rural women have below normal BMI. Up to 85% of women are anemic, and the maternal mortality ratio is 312. That's why the goal of the study is to identify the characteristics and interrelations of soil, plant, and human nutritional health in Jharkhand's rice systems to better address the issues of adult female malnutrition. So I'll be covering 2 parts. The first is a quick overview of some of the minerals present in the soil, rice, and humans. And the second is the assessment of the interlinkages between these 3 mediums. So within the scope of this assessment, minerals were categorized into 4 categories. So we have the essential mineral micronutrients, the essential mineral macronutrients, the potentially beneficial trace elements, and the toxic elements. So while I'll be reporting each mineral's essentiality to plants and humans and their functions briefly, I'll also be reporting some thresholds or baseline values that allow us to know what the optimal concentrations are in each medium. For soil, they will be reported in terms of Mehlich 3 in parts per million. For rice, they will be reported in terms of maximum permissible level whenever the information is available. Otherwise, the recommended dietary allowance, and that's in milligrams per day, and the maximum tolerable intake, which is also reported in milligrams per day. And then for human hair, and I'll explain why human hair is important in such studies, they'll be reported in terms of reference ranges, and they're either in the 5th to 95th percentiles or the 10th to 90th percentiles, and these just represent the normal range. So all all the minerals are reported in this table and the next 3 tables. Behind these 4 tables went a major probing of the literature that lasted for months and resulted in 70-page literature review. But so I'll be reporting. I'll be reporting just the first 2. But I encourage you to really look at these values. And if you disagree with them, please do let me know at the end of the talk, and we can discuss them. So these were really the best I could find from those 4-month-long literature review. So boron is essential to plants and humans. Boron is essential to plants, and it's suspected to be beneficial to humans. It hasn't been really conclusively proven so. In plants, it's important for protein formation, amongst others. And for humans, it's suspected to be important for the metabolism of minerals. The soil malic 3 deficiency critical level is 0.4. The toxicity critical level is 5. There is no RDA because it hasn't been conclusively proven to be essential, and the MTI is 20. Again, RDA recommended dietary allowance, MTI is maximum tolerable intake. And then the human hair reference ranges are 0.26 to 1.87 reported for a study conducted in Canada, and 0 to 3 in Poland. Cobalt is essential to humans and hasn't been conclusively proven to be essential, at least not directly to plants, because it's the nitrogen fixing bacteria that need cobalt. And for humans, it actually constitutes the backbone of vitamin B12. The soil malic 3 deficiency or toxicity critical level is 4. The RDA, there is no RDA. The maximum permissible level in rice grains is 0.5, and these are the reference ranges. And so you can look at these. I won't be covering them. I'll just say that chromium is essential to humans. Copper, iron, manganese is essential to both. Molybdenum is essential. Molybdenum, nickel is essential to both plants and humans. Selenium is only essential to humans, and our intake of selenium really relies on the inadvertent uptake of selenium by plants. Titanium is beneficial to plants. Vanadium beneficial to plants and suspected to be essential to humans. And zinc is essential to both plants and humans. Now, the macronutrients, all of them except for sodium, are essential to both plants and humans. And then for the toxic elements, aluminum, arsenic, barium, cadmium, lead, and strontium were all assessed against these sort of baseline values. All right, sample collection. In 2015, soil samples were collected from randomly selected rice fields across 7 districts in India, in Jharkhand, India. In 2018, rice and human hair samples were collected from the female individuals in the households that have been cultivating and subsisting on the rice fields that we collected soil samples from. Additionally to the field rice that was collected, we also collected the government subsidized public distribution system rice, because it often constitutes the second most consumed rice in the household after the one that's grown by these households. And so why hair? Why are we assessing hair for nutrition status? Well, hair has many advantages over the assessment of blood or urine. First, it's non-invasive. Second, it's a long-term bioindicator, so it's able to reveal retrospective information about a human's nutritional status over an extended period of time. And third, it's easy to store and to transport. So soil samples were subjected to against a set of physical, biological, and chemical health indicators. The physical indicators included aggregate stability, available water capacity, surface and subsurface compaction. The biological indicators included organic matter, active carbon, soil respiration, and soil protein. And the chemical indicators included pH and all of the elements that we discussed earlier. For the soil, the samples were assessed for available content using Mehlich-3 extraction. For the rice and hair, they were analyzed for total, and they were quantified using the ICP-OES. In addition to these elements, we also analyzed phytate. So phytate is it's a natural compound found in many cereal grains. It's an anti-nutrient that inhibits the absorption of iron and zinc. So it interferes with the absorption of iron and zinc in the gut. And that was analyzed using a commercially available kit called Megazyme. And the procedure basically includes briefly the extraction of phytate and then undergoing a series of enzymatic dephosphorylation reactions, adding a color reagent and quantifying it with a spectrophotometer.
So these soils were sampled in India and then brought here, and the analysis was done here, including the phytate analysis?
Yes, the phytate was done for the rice.
Okay.
Yeah. So in order to interpret the results, soil and or rice samples were translated into scores of 100 or 0 on the basis of sufficiency or deficiency toxicity when the thresholds were available, as I pointed out in the tables. Otherwise, we basically computed the percent contribution of the nutrients in the rice to their RDAs, the recommended dietary allowance, when applicable. In hair, as you saw, there was a huge lack of agreement amongst the reference ranges. And so we crudely compared our samples to the reference ranges. So were they above, below, or within at least one reference ranges that was found in the literature. All right. So the results of the soil health analysis revealed that 95% of our soil samples were below the wet aggregate stability critical level. 7% were below the available water capacity critical level. 79% and 57% were below the surface and subsurface compaction critical level. So a big proportion of the soil samples were suboptimal as it relates to soil physical health. These results imply that the current physical state of Jharkhand soils is largely a result of the degradative influence of the popularly adopted management practices of puddling for paddy rice production. So which destroys the structure of the soil. Similarly, the results for the soil biological health analysis showed that 93% of our soil samples were below the threshold for organic matter content, and all of the soil samples were suboptimal as it relates to active carbon, protein, and respiration. And these results suggest that Jharkhand paddy soils are extremely biologically poor, which may be factored by the continuous monocultured and undiversified rice cultivation. Okay, so now looking at the soil, the mineral concentrations in the soil. So we find that 51% of soil samples were deficient in boron. 65% were in excess barium. 28% were deficient in calcium. Where is calcium? 12% in excess cobalt. 9% and 3% deficient and in excess copper, respectively. 16% in excess iron. 86% deficient in potassium. 30% and 49% deficient and in excess manganese. 7% in excess sodium. 2% in excess nickel. 95% deficient in phosphorus. 5% in excess lead. 37% deficient in sulfur, and 86% deficient in zinc. And these results suggest that Jharkhand soils are constrained by limitations in the content of essential elements and by toxic, by excess toxins. So we also see that Jharkhand soils are constrained also by the chemical state. So moving on to rice, we can make three observations. The first one is that field and PDS samples contributed to the micronutrient RDA on average between 1 and 3% for calcium, 185 and 274 for chromium with no potential toxicity, 77 and 78% for copper, between 11 and 19% for iron, 19 and 17% for potassium, 42 and 29% for magnesium, 201 and 78% for manganese, with 2% of field rice samples exceeding the MTI, 260 and 284% for molybdenum, with no samples exceeding the MTI, 0.46 and 0.32 for sodium, 68 and 62% for phosphorus, and between 13 and 25% for zinc. And when we considered the rice zinc concentration critical levels, more than 90% of samples were considered deficient. And finally, about 80% exceeded the lead critical level. Okay. The second observation we can make is that field rice samples were significantly higher than PDS rice samples in calcium, potassium, magnesium, manganese, and zinc, all of which are essential macro and micronutrients. So again, the field rice samples had significantly higher micronutrient and macronutrient content than the government subsidized public distribution system rice. The third observation we can make relates to the phytate to mineral molar ratio, which was computed to predict the inhibitory effect of the anti-nutrient phytate on the bioavailability of zinc and iron. And it showed us that almost all of our rice samples were suboptimal by exceeding the threshold. So that means there's more phytate to iron concentrations. So these results suggest that rice consumed by the women of Jharkhand are by and large fulfilling the micronutrient RDA to some acceptable degree, with the large exception of critical micronutrients, iron and zinc. Those are big ones. And this may be due to the phytate to iron and zinc ratios. To a lesser degree is the rice fulfilling the macronutrient RDA. And here, not much can be said. Using our crude method, we found that the elements that were within at least one of the reference ranges were calcium, cobalt, chromium, copper, nickel, sulfur, titanium, vanadium, and zinc. The elements that were above at least one of the reference ranges were aluminum, barium, cadmium, lead, strontium, potassium, iron, magnesium, manganese, and sodium, and phosphorus was the only one below. It is important to keep in mind, however, that this categorization may or may not indicate existing constraints due to the multitude and often contradicting reference ranges, as we saw earlier. And that is why human hair analysis has yet to become widely adopted, because every study is sort of expected to define its own healthy population and to be used as a baseline in their studies. So the question is, do the constraints in the soil translate to constraints in the rice and the hair? Well, let's see. Some of these results suggest that the content of some elements in rice and subsequently hair contribution to human nutrition may be dependent on their respective content in the soil. Like a good example is chromium and molybdenum, but not all. A good example of it not corresponding well is iron. So the excess iron in the soil may not be associated with similar trends in the rice, as evidenced by its low contribution to the RDA, relatively low. And this could largely be due to the bioavailability dilemma. But it is parallel to human hair levels. Similarly, 95% of Jharkhand soils are phosphorus deficient, yet the rice that grows on these soils contribute to 68% of the RDA, but also paralleled with hair. Finally, 5% of samples exhibited excess lead, while 80% of the rice grown on these soils have excess lead. So why is that? So in any case, we should really take these results with a grain of salt because of this phenomenon. You know, are the critical levels or reference reagents even reliable? Are they valid? Are they compatible? In the case of lead, is there another source of contamination? How about the other sources of mineral in the diet? I mean, surely they don't just eat rice. How about mineral bioavailability? So that's why why further analyses are warranted to verify these connections. We used in our preliminary step, we used Pearson's correlations to verify this. So I don't expect you to, oops. I don't expect you to be able to read. I mean, I'm going to walk you through these results, but if you would like to follow along, soil is in the x-axis, hair is in the positive y, and rice is in the negative y. The results suggest that soil physical and biological health indicators are significantly correlated to rice and hair mineral content. There are associated benefits of higher physical soil health to humans via increased availability of essential nutrients and decreased availability of potentially toxic elements. We also found that organic matter is beneficial not only in its supply of nutrients to plants, but also in its association with suppression of potential plant and human toxicity. We also found associated benefits of higher soil biological health and specifically soil protein content on the reduction of lethal toxins such as lead in rice. In general, these results suggest that soil physical and biological health indicators are quite correlated with rice and human health nutritional status. All right, so here we have, and again, this is for those who would like to follow along. We have soil in the x-axis, hair in the y, and rice in the z. The results suggest that an increase in toxic elements in soil is associated with an increase in other toxic elements in rice. And we found that for soil, aluminum, arsenic, and cadmium, with rice, barium. Possible antagonistic effects between soil and rice toxins, and that was the case for soil cadmium, arsenic, aluminum, as they relate to rice, lead. Higher micronutrient content in soil is associated with decreasing toxin concentrations in rice, and we saw this with soil boron, iron, potassium, and molybdenum, and selenium with rice, lead. Toxicity in rice associated with the same and different toxins in hair. But in general, the soil to hair trend was unclear, given the indirect relationship via nutrition. The direct link between hair minerals and soil physical and biological health indicators were to a certain degree more apparent than with soil minerals. So in conclusion, human health is a main pillar in sustainable agriculture and should be at the forefront of the agricultural systems agenda. It is known that the resources that shape our environment play an immense role in determining our health as individuals and communities, specifically the soil, which forms the foundation of life. Jharkhand soils are characterized by poor health in all aspects. The soil status was to a certain degree reflected nutritionally on rice and humans. And further research is warranted in this discipline. So the next steps, so what I presented was more of the preliminary statistical analyses. The next steps will include some multivariate analysis and perhaps the path analysis and lasso regression. Also an in vitro iron bioavailability assessment, studying how the socio-demographic and lifestyle affect the soil, rice, and human health, and conducting some spatial analyses. In other words, are soil toxicity and human toxicity co-located? And is it a result of proximity to industrial facilities? I'd like to acknowledge the NSF and the Tata Cornell Institute being my two funders, and I'm happy to take any questions. Yeah, we can start with you.
You have controls for this soil sample?
Controls?
Yeah, like enriched soil versus the deficient soil, and then you correlate the nutrition deficiency with the healthy population.
No, we reverted to just the literature to see what are some the thresholds that other the general consensus was on for each element. Yeah.
So Fatma, what was the kind of range in pH of those soils?
They were between 4.5 and 5.5. So pretty acidic. Yes.
You had the two sources of rice from the farm itself where you sampled, and then from the government.
Correct.
Right? And so did you look at the proportionality of those in the diet and try to use that to translate in what you've got, what you're finding?
Yeah. No, not for this step. We don't know the proportions. We did as part of a survey. We did ask them in what quantities or how often do they eat, or how many times do they eat rice per day. But we didn't really differentiate So we did ask which was the dominant, and some of them said, all of them said the dominant source is is from what they grow, and then if they run out, then that's when they revert to the PDS rice. Yeah.
Is the homegrown rice processed kind of similarly or very differently from the commercial rice?
Yeah, so what they do is they take their rice and they go to the village mill and they mill it. I would assume that the government also, I don't assume to have any I don't think they would be different. So, but I don't have such information.
It does sort of indicate that the rice that's part of the distribution system is of lower quality.
Yeah.
Right. So that may be because it's being grown in other parts of the country.
Yeah.
Probably further west in the Gangetic Plain.
Yeah.
So perhaps those soils are in worse shape than the ones in Jharkhand.
Correct. And you know, when the PDS rice is really just about having enough stocks. You know, it's just trying to maximize yield. And so really, if any type of fertilization that would go into the rice that's acquired by the PDS system, or the PDS, is really those that have been fertilized to maximize yield and not really the micronutrient content.
One other question about the soil health indicators. You indicated that were the critical levels. So was my understanding that the soil health indicators have not been established for India so far. So were you using the US criteria?
Correct. So typically, what I did for my master's is I created my own scoring. So I adapted the framework and created my own, and that's because I was able to. I had a large enough sample. So I had 200. My sample size was 255. In this case, I can't. Yes, they were normally distributed, but I can't. My sample size is too small for me to just assume that this is everything, you know, and just base my create my own scoring functions. So that's a limitation.
But that may be a potential.
Correct. Absolutely. Absolutely. Yes.
So maybe I missed this, but are you analyzing? I understand the issue with the information about what the contents in hair samples mean. But are you looking at looking at correlations with uptake, because you have quantitative information about the amount in the hair? So I guess I'm asking, like, it would be useful information to just relate that to the rice and the soil.
Correct. And that's what was
qualitatively, even though you can't say
qualitatively
what's healthy and what's
right. Exactly. And that's what we did in the Pearson's correlations, and hope. So this is just really the preliminary step, and then we'll take it more to doing some multivariate analyses. Okay. I think, sure.
Could you consider for the next step to see a water source
Water source?
Yeah, like the chemical properties in water.
Yeah, I think, I think that another PhD student in the Tata Cornell, I think, was looking at water. So yeah, that is an important, it is important. But its effect is, so drinking water, right? But irrigation water it's rain-fed. These are rain-fed systems. So it wouldn't be through the rice. It wouldn't be through the rice intermediate. So it would be just direct consumption of water, drinking water. Yeah. Yes.
Your talk, very interesting topic and results. I'm wondering when you're looking at your literature review and considering your human health indicators, did you come across any evidence for like hair relation to other biomarker potential? Like, what do we know about hair versus urinary biomarkers and blood biomarkers?
Yeah. So what I do know about urine and blood is that the issue that researchers that use these bioindicators is that they change by the hour. So in the case of blood, it's not really reliable because it changes every hour, and the same for urine, changes every day. So those are the main constraints. Yet most researchers still use that because we know what the thresholds is. In hair, the limitation is that each study is sort of defining its own normal reference range, and so there is no agreement, and that's why it's not really widely adopted. Does that answer your question?
I like the volatility of the other indicators to some extent, but I just sort of love to know if anyone's working on sort of large enough data sets that, given the volatility of the for mycotoxins, which is my area of interest, the urine is volatile and the blood is much more stable for some consumption over a couple months, as opposed to a couple of days. I would kind of be interested in knowing how we might get panels of data that might allow us to kind of see how the three biomarker systems might interact, behave, and kind of start getting a feel for, taking an interest in the variants as well as the mean, and then, I can see the beauty of hair as a assay system.
Yeah.
Oh my God. But just to know if, could we kind of move towards having acceptance of different things for different purposes?
Yeah, I haven't come across such studies, but it's not to say that they don't exist. But it's something that you raise a really interesting topic. Yeah. Yes.
We did analysis of hair from questions, your samples or someone else's in this
Probably
study.
mine. Yeah.
But there's a couple people that sent hair to us, and we noticed that the hair at the end was higher and more dense than it was at the top of the head. And some of that came from the fact that as they brush their hair, they slip off a lot of the cells at the ends because it gets brushed more. And so the concentration varied. It was more dense at the base of the end of the hair than it was near the scalp.
Interesting.
So there needs to be some standardization of what hair sample you collect.
Yeah, no, for sure. Well, when I collected these hair samples, I bought a Barbie and cut off a strand to show them how much we would need and try to keep it uniform. But you run into, so we got a lot of our female participants refused to give us hair samples out of fear of superstitious beliefs. And that's why my hair sample size was smaller than the rice and the hair. And some of them said, no, I'm not gonna cut off any hair for you, but I can give you the leftovers from my comb. And so we accepted that. But in general, we were trying to keep uniformity of how much we want from the tips. But unfortunately, it wasn't always successful, just because of the participants' willingness to give their hair samples.
All right, so maybe we should.
This has been a production of Cornell University, on the web at cornell.edu.